Okay. Well, it's a little bit after 7, so I'll go ahead and introduce our speakers so that we can get things started. So thank you everyone who's called in. I see we have several participants called into tonight's meeting. Tonight is our Evenings with Genetics seminar on adult neurogenetic conditions. So for anyone who's not familiar with Evenings with Genetics, this is a series that's sponsored by the Department of Molecular and Human Genetics at Baylor College of Medicine and Texas Children's Hospital. And my name is Amanda Gerard. I'm a genetic counselor at Texas Children's Hospital, and I'm on the planning committee for Evenings with Genetics. So I'll be helping to moderate tonight's seminar, and we're going to hear from 2 provider speakers tonight, 1 physician and 1 genetic counselor in the Neurogenetics clinic, and then that'll be followed by about a 15-minute Q&A session. So you can enter questions for the speakers using the Q&A function in Zoom at any point during the seminar. And we'll be able to see these, and we can answer questions at the end during the Q&A session after both of our presenters have spoken. And I'll also give you an opportunity to unmute your microphones at the end of the seminar as well to ask questions during the Q&A portion. And then towards the end of the seminar, also keep an eye out in the chat for a link to a survey evaluation. We really appreciate it if all of our participants are able to complete that to help give us some feedback and ideas for future seminars. And so I'll go ahead and introduce both of our speakers, and then we'll let them get started. So we have Laurie Robak as our physician speaker. She's a medical geneticist who attended the University of Rochester School of Medicine, where she obtained her MD and Ph.D. She did her combined pediatrics and medical genetics residency at Baylor College of Medicine, and currently she's an assistant professor of molecular and human genetics, studying the genetics of Parkinson's disease and other neurodegenerative conditions. She's the director of the Adult Neurogenetics Clinic at Baylor College of Medicine. And our genetic counselor speaker is Jamie Fong. She's a genetic counselor and assistant professor in the Department of Molecular and Human Genetics at Baylor College of Medicine. She's worked as a genetic counselor since 2008, providing counseling and support to families at risk for inherited conditions. At BCM, she provides genetic counseling to people either with or at risk for Alzheimer's disease, Parkinson's disease, frontotemporal dementia, ALS, cerebellar ataxia, and related conditions. She's a co-principal investigator of an interview study funded by the BCM Office of the President to investigate the words Spanish-speaking Mexican Americans used to describe precision medicine for Alzheimer's disease. And she also facilitates a telephone support group for adult children of people with frontotemporal dementia. She lectures frequently about genetic counseling and dementia care, and she holds a master's degree in genetic counseling as well as a bachelor's degree in molecular and cell biology. And so with that, I will turn things over to our speakers.
Thank you so much, Amanda, for introducing myself and Jamie. We work closely together studying the genetics of neurodegenerative conditions. So neurogenetics is a field that seeks to understand the genetic basis of brain disorders. And really what we'd like to do is to not only improve diagnosis, but use genetics in order to find potential treatments and determine risks for other family members. Okay, so when should you see a neurogeneticist? So the reasons that we've come up with are if you've got a known or suspected neurologic disorder that may have a genetic cause, if you have family members with a neurologic disorder and you want to understand your risk, even if you yourself don't have that neurologic disorder. And if you got genetic testing results through either a different doctor or through some other sort of testing mechanism and that have implications for a neurogenetic condition that perhaps were something that we're not expecting, you want to discuss what they might mean for your potential health. There are hundreds of types of neurogenetic conditions that can be seen in adults. We're going to focus on some of the more common ones today. Jamie has expertise in dementia and Alzheimer's disease, and she'll speak first about that. I study Parkinson's disease, so I'll talk about that. And she will talk a little bit about the ataxias while I will talk about ALS. But this in no way encompasses the breadth of everything that we could see in our genetics clinic. We could see individuals who have Huntington's disease, frontotemporal dementia, which I'll talk a tiny bit about, dystonia or difficulties with muscle tone, spinal muscular atrophy, hereditary spastic paraplegia, and the list is really quite endless. But if there's a neurologic component to a genetic disease, we would be more than happy to see any patients with such conditions. And I'll turn it over to Jamie now.
Thanks, Laurie. So what is neurodegenerative disease? Next one, arrow down. So there are different definitions, but I think on a fundamental level, we would define neurodegenerative disease as progressive decline from a previous level of functioning. So it recognizes that a person was operating at a certain level of cognition and thinking, and there are changes that now interfere with daily living. Next. And this includes the profound and persistent disruption of work life, home life, and social life. Next. And Alzheimer's disease is just one type of neurodegenerative disease. Next. So what is Alzheimer's disease? I think we have a video to share that I think encapsulates Alzheimer's disease quite nicely.
What is Alzheimer's disease? Alzheimer's is a slow, fatal disease of the brain affecting 1 in 10 people over the age of 65. No one is immune. The disease comes on gradually as 2 abnormal protein fragments called plaques and tangles accumulate in the brain and kill brain cells. They start here in the hippocampus, the part of the brain where memories are first formed. Over many years' time, the plaques and tangles slowly destroy the hippocampus, and it becomes harder and harder to form new memories. Simple recollections from a few hours or days ago that the rest of us might take for granted are just not there. After that, more plaques and tangles spread into different regions of the brain, killing cells and compromising function wherever they go. This spreading around is what causes the different stages of Alzheimer's. From the hippocampus, the disease spreads here to the region of the brain where language is processed. When that happens, it gets tougher and tougher to find the right word. Next, the disease creeps toward the front of the brain, where logical thought takes place. Very gradually, a person begins to lose the ability to solve problems, grasp concepts, and make plans. Next, the plaques and tangles invade the part of the brain where emotions are regulated. When this happens, the patient gradually loses control over moods and feelings. After that, the disease moves to where the brain makes sense of things it sees, hears, and smells. In this stage, Alzheimer's wreaks havoc on a person's senses and can spark hallucinations. Eventually, the coordination, and in the very last stage, it destroys the part of the brain that regulates breathing and the heart. The progression from mild forgetting to death is slow and steady and takes place over an average of 8 to 10 years. It is relentless and, for now, incurable. Helping your family, friends, and neighbors to better understand Alzheimer's will reduce stigma, improve care, and even help the fight for a cure. To do your part. At www.aboutalz.com.
That was an overview, and I hope that you can hear this. Apologies for the tech issues. You may be wondering, I've heard of Alzheimer's, I've heard of dementia. Are they the same? Are they different? I want to show you now just a short video describing the two.
What's the difference between Alzheimer's disease and dementia? Perhaps you've been confused by these terms in the past, or mistakenly thought they were the same thing. Well, you wouldn't be alone, because knowing the difference between
Jamie, I think your video is pausing. You may just want to verbally state what
asthma.
this says.
Shortness of breath is not a specific disease. It can have many different causes, and asthma is just
Sure.
one of them.
Okay.
Likewise.
Yeah. So, yeah.
It's a combination of
Basically, they're not the same thing. They are distinct. Dementia is an umbrella term. Alzheimer's disease is a type of dementia. It's worthwhile. It can be worthwhile to investigate any potential type of dementia, clarifying whether it's Alzheimer's disease or a different type of dementia, if only because there can be behaviors or maybe medicines might be more amenable to someone with Alzheimer's disease versus a Lewy dementia or Parkinson's disease with dementia. So yeah, I regret that's not available to you. In terms of the question surrounding, well, how likely could Alzheimer's? In reality, genetic Alzheimer's is actually rare. Fewer than 100 cases being caused. If we take Alzheimer's cause, there are very distinct features that it has. It tends to appear at very young ages, at or under age 40. It also occurs in families with many people with symptoms on one side of the family or the other, not usually because your parents are related to each other by blood. And those individuals with symptoms tend to occur in back-to-back generations. The features besides memory problems that we see in purely genetic Alzheimer's disease, and this can include history of seizures.
Jamie, I'm very sorry to interrupt, but you're breaking up pretty badly.
Other features.
Jamie, I'm sorry to interrupt you, but you're breaking up pretty badly.
Okay. Yeah.
Should I switch over to my part of the presentation and then maybe you can reboot while I do that?
Go ahead.
Okay.
Yeah.
Sorry, everybody, about this.
Okay.
We're going to switch over to Parkinson's disease, and then we'll switch back to Alzheimer's disease when Jamie has a chance to reboot. So while Jamie's rebooting, we're going to talk a bit about Parkinson's disease. And here is a word diagram of Parkinson's in the shape of the brain with a number of words that people have associated with Parkinson's disease. We're going to talk a little bit about what Parkinson's disease is and what genes can potentially cause PD, as I'll refer to it from now on. So PD starts out gradually and it's progressive. And there's really about a 20-year prodrome that can happen, which means all the stuff that happens before you're physically diagnosed with the disease. But diagnosis happens when you start to exhibit several of the following features. One is a tremor at rest. When you're sitting at rest and your hands start shaking, slow movements, and that is known as bradykinesia, rigidity or increased muscle tone, and then gait impairment or postural instability. And some people can have what we call tremor predominant, where they have more of the tremor problems, but not so much the posture issues. And other people can have more of the gait impairment, postural instability subtype, where they have less tremor issues. Preceding PD, and then also symptoms that can become worse over the course of PD include dysautonomia or difficulty regulating temperature. There's a restless leg disorder that can occur before PD as well, depression, and then late in the disease, you can get cognitive impairment and dementia. There are treatments available and they do work. The problem is that they rely on your neurons to be functioning. And over the course of PD, what happens is your neurons start to die, and the more and more cell death you have, the less these treatments work. And so for that reason, because Parkinson's disease remains incurable and progressive, even though we do have these treatments that work pretty well for a number of years. So it's the second most common neurodegenerative disease after Alzheimer's, and Jamie will talk about that in a moment. And it's the prevalence, which means how many people in the population have Parkinson's disease, increases with age. So roughly 1% of the population around age 65 will have Parkinson's, and about 5 times as many will have that at age 85. For reasons that are unclear, males tend to be twice as likely to be affected as females. We know that there are both environmental and genetic risk factors. I'm really only going to be talking about the genetic risk factors today, though. So this is showing you what the affected part of the brain looks like. So what we're showing you on the right-hand side is a cross section through the head. And these are the eyes up here, and then the ears are off to the side. And in the middle is what we call the midbrain. And if you blow up a photograph of the midbrain, you have these neurons that are all these little black dots here that appear dark. And that's called the substantia nigra, which means black substance. And, for obvious reasons, when those neurons start to die, you actually have less of that black pigment left. And that's what it can look like in Parkinson's disease. On the right-hand side is showing you a number of neurons. This is just a very, very close look at the neurons in the brain. And all of these brown labels here are basically alpha-synuclein, which is the type of protein that builds up in Parkinson's disease. And it can develop into these little bodies, which we call Lewy bodies, which is also another pathological hallmark of Parkinson's disease. So it's really interesting, the history of PD, because really one of the initial diagnostic criteria for PD was that it was thought not to be genetic. And in fact, the clinical diagnostic criteria said if you had a family history of PD, you couldn't have PD. And so I'm showing you the international sort of diagnostic guidelines that are now outdated for Parkinson's disease. And if you look at the exclusion criteria, which means if you have any of these criteria, you don't have PD. If you have more than one affected relative, you were considered as not having PD. We now know that that's not to be true. At least 10 to 15% of people with Parkinson's disease have at least one family member with Parkinson's disease. So the types of PD genes that we can have are of several different types. There are some genes that almost always cause PD. So if you look over at the left-hand side of the graph, these are genes. This shows you the effect sizes as you go up the graph, and then how rare they are and to how common they are as you go from left to right. And so genes that always cause Parkinson's disease include genes like alpha-synuclein, which kind of makes sense because alpha-synuclein is the pathological hallmark of PD, or it's the protein that's always involved in cell death and PD. At the other end, where we have very, very common variants that have teeny tiny effect sizes, there are at least 90 different genetic changes that go in these categories, where by themselves they have very small influence on PD risk. But we're now starting to understand that if you put them all together into what we call a genetic risk score, that they may have actually a larger impact on PD. And then somewhere in the middle, we've got genes that increase your risk for Parkinson's disease, but aren't sufficient in and of themselves. And one of the examples is GBA. And I will talk about each one of these, categories in my talk. In practice, when we look at people in the neurogenetics clinic, we can really only test right now for genes that almost always cause PD and genes that sometimes cause PD. The genetic risk variants with very small effects it's still a work in progress, and we're still trying to really understand what that means for an individual person. There's a lot of research right now trying to understand that. So Parkinson's disease risk genes are autosomal dominant. So that means if you've got an affected parent, and here it's the father, it could be the father or the mother, and the mutation is shown here in purple, 50% of that person's children can be affected with the gene change. So genes come in pairs, and there's only one mutation, and that mutation can be passed down to about half the children. So that doesn't mean that if you've got 2 kids who are unaffected, you will then have 2 kids who are affected. It means for every single pregnancy, there's a 50% chance of the pregnancy being affected. And so you could have no affected children, or you could have all affected children. It's really a coin toss for every pregnancy. And an example of an autosomal dominant gene is alpha-synuclein, or what's called SNCA. So SNCA gene encodes for alpha-synuclein, and I mentioned before, it forms the Lewy bodies that are found in the substantia nigra, which is the black portion of the brains of people with Parkinson's disease. And we know that if you've got too much alpha-synuclein, then we have a chromosome duplication that causes PD. And this is kind of what it looks like, where you've got this pink and green region that ends up being duplicated here. And so you end up having too much alpha-synuclein, and that can cause Parkinson's disease. However, we also know that if you've got misspellings in the alpha-synuclein gene, you can get Parkinson's disease. Shown here on the right-hand side of the graph is individuals with either a point mutation, which is a misspelling, or a duplication. It's 100% penetrant, which means every single person who has one of those changes will get Parkinson's disease by around age 75. You can also have, instead of a duplication, a triplication. And individuals with triplications get Parkinson's disease even earlier. And there's even people who have 4 or 5 different copies, and they get Parkinson's disease quite early. Another very famous Parkinson's disease gene is Lurk2. Lurk2, the protein, is a risk factor for PD, and it's very common. It explains about 10% of familial PD. So that means if you've got Parkinson's disease and a family history of Parkinson's disease. And then what we call sporadic, which means you don't have a family history. And the different gene mutations you see are pretty much uniformly 100% penetrant, which means almost everyone who has these genetic risk factors will get Parkinson's disease. But we know that in certain backgrounds, so if you are of Norwegian descent and you've got this particular mutation called G219S, you'll get Parkinson's at a later age, and not everyone will get Parkinson's. And the reason for that is not entirely clear yet, but we think it might be related to either environmental factors or something like the genomic risk score, which I talked about earlier. Another important concept in Parkinson's disease risk is this concept of incomplete penetrance. So that means that not every single person who has a gene change will get Parkinson's disease. So shown here in blue are the individuals who have a gene mutation. So this individual passed it on to 50% of their children, which is what you would expect. And then this individual had a child who was then affected. My apologies. But what it turns out here is that this individual had the gene mutation and then passed it down to their child. And we know this because their grandchildren have the disease, even though their child does not. And that's what we call incomplete penetrance, where even if you got a gene mutation, you don't actually get the disease. And a prime example of this is GBA, which is another famous PD gene. So GBA mutations cause a different disease, which is a lysosomal storage disorder called Gaucher disease. So it's autosomal recessive. So in contrast to what I showed you for alpha-synuclein, where you need 1 gene mutation, you need 2 gene mutations in order to get GBA. In order to get Gaucher disease that's related to GBA. Gaucher disease is associated with bone problems, a big liver and spleen, and then low red and white blood cell counts. And also a small percentage can have some significant lung involvement as well. What's interesting is that if you've got Gaucher disease, you have an increased risk of PD. So if you have Gaucher disease, which means you've got 2 GBA mutations, you have a 20-fold increased risk of Parkinson's disease. And what that means is that if you're age 65 and you've got Gaucher disease, instead of the 1% risk in the whole population, 20% of people with Gaucher will have PD at that age. But that means 80% of people with Gaucher will not have PD at that age. And so there's got to be something else that's influencing PD risk. Could be potentially other genes or environmental risk factors. We're trying to explore this more as a genetics community. The other interesting things that comes out of this is that GBA can also act in an autosomal dominant fashion. So this means if you've got 1 mutation, you have a 5 to 8-fold increased risk of getting Parkinson's disease. So about half of what your risk is if you have Gaucher disease. So that means at age 65, your risk of getting Parkinson's disease is 5 to 8% as compared to 1% for the general population. But most people with a GBA gene mutation will not have Parkinson's disease at that age. But the risk does increase with age for whether you have Gaucher or whether you have one GBA mutation. And then just to bring home the point about autosomal recessive, because there are other genes that can cause Parkinson's disease that are recessive. Autosomal recessive means that you need 2 mutations, as I mentioned previously, in order to get a disease. So these are 2 parents who each carry 1 mutation, and they have 1 affected child who has 2 mutations, 2 of the 4 children who are carriers, which means they are capable of passing it on to their children, and then 1 child who's neither a carrier nor affected. And the 3 genes that cause autosomal recessive Parkinson's disease are PRKN, which is also known as Parkin, PINK1, and PARK7, which used to be called DJ1. And what we know about mutations in these autosomal recessive genes is that people tend to get PD earlier than in the autosomal dominant forms. And then this is pertinent to anyone in the audience who has a family history of Parkinson's disease, or maybe they themselves have early onset Parkinson's disease. So this is a study that I published in conjunction with Jim Lubsky and Josh Schulman here at Baylor. We looked at people who had either a 1st, 2nd, or 3rd degree relative with PD. That means that they had an affected parent or grandparent or aunt, and we looked at different genetic changes. And basically, we found a genetic cause in about 20% of people with Parkinson's disease. There's another paper that came out looking at early onset Parkinson's disease. That means they were diagnosed before the age of 40. And the detection rate there was 11%, although it was published a little bit earlier than ours. And so they were missing some of the more important gene changes that have been discovered since. So I'm sure that number is probably even higher. And so if you are considering getting genetic testing for PD, what might be the reasons? Or how do you actually get genetic testing for PD? It has to be ordered by a doctor or a genetic counselor, depending on what state you're in. Can be ordered by a neurologist or a geneticist. And there's different types of tests. There's a panel, which means that tests some of the main PD genes, just maybe 20 or so of them. There's an exome test, which looks for spelling tests in all 22,000 genes in our genome. And then there's also a chromosomal microarray, which looks for missing or added pieces of chromosomes. A couple of things to note, though, is that insurance does not always cover testing. One nice thing that we have is that some companies do offer sponsored testing, meaning that it's currently free of charge. Right now, there's a certain company that is offering it free of charge, but basically they're doing research on us, the physicians who are ordering these tests, and that's what they're getting out of it, because that will help with their profit margin later on. Jamie, are you ready to move on to and go back to your Alzheimer's section?
Sure, yes.
Okay.
I believe so.
So just give me a moment to go back to the correct slide, and then I'll keep sharing my screen if that's okay.
Yes, thank you.
Okay, and then maybe you want to talk about the difference between Alzheimer's disease and dementia, because I think that's kind of where we left off.
Oh, sure. Okay, that's where it kind of.
Oh no, geez Louise. Sorry, everyone. It somehow is not showing up on the right slide. Here we go.
Yeah, so I can talk. Yeah, so these videos just illustrated that what Alzheimer's disease was. It's I think it is a myth that all memory problems are dementia, or that all dementia consists of memory problems. Next slide. I think the key difference between Alzheimer's and dementia is that dementia is an umbrella term. Alzheimer's is just one type of dementia. Whenever we invoke the term Alzheimer's disease and we think about our family histories, often the question arises, is this inherited? Could this be passed down in a family? And it's important to keep in mind that most instances of Alzheimer's disease is not genetic. Genetic Alzheimer's is rare. Fewer than 1 in 100 families or cases, excuse me, is actually due to a single gene cause. Next slide. So, so monogenic Alzheimer's disease, or Alzheimer's disease caused by a single gene cause, tends to occur very young, at ages under age 40. People are often very impaired and potentially needing help with all activities of their daily living by their 60s. It occurs in families where there are many individuals with Alzheimer's disease, or sometimes it's just called dementia or senility. These individuals are on one side of the family or the other, not usually both, unless your parents are related to each other by blood. And also these individuals are found across back-to-back generations. Single gene caused Alzheimer's disease may also feature symptoms in addition to the classic symptom that we think of and that you might have heard about in the video, and that is memory problems. But additionally, genetic forms of Alzheimer's disease can feature symptoms like seizures, spasticity, which is tightening or stiffness of the muscles. And the genes that we invoke when talking about Alzheimer's disease caused by a single gene mutation or variant are genes that go by the abbreviations of APP, PSEN1, and PSEN2. Again, those are rare. Next slide, please. So most instances of Alzheimer's disease is what we would call multifactorial. It arises from a combination of genetic and environmental factors. And there's no one gene that is responsible entirely for disease. So again, this is in contrast to the families that face a purely genetic form of Alzheimer's. Next. And so many genes are involved, each with a small effect or small impact on the development of disease. Next slide. And one way I like to think about it is a jar analogy. I don't know if you've seen or heard this before, but take this jar, and if we arbitrarily just draw a line there that says that that's the threshold for Alzheimer's symptoms, and you think about genetic Alzheimer's disease, this marble in this jar represents the single genes, the APP, PSEN1, PSEN2, that when harboring a mutation or a change leads directly to Alzheimer's disease. You can see that the size of the marble, it is enough to cross that threshold. And so what is multifactorial Alzheimer's disease? So we take the same jar with the same threshold, and instead we've got more than one gene, as represented by the pink marbles. They're of smaller size than the blue marble in the picture next to it. And then we've got little marbles that we've labeled with E to represent environmental factors. So you can see that it takes a group of those factors in concert to push someone over the threshold for symptoms. And so what is sporadic Alzheimer's disease? So again, take the same jar, but instead we've got many more genes that are much smaller than the genes in the other jars. And we still have even more environmental factors. Again, it's the constellation of all those factors that pushes someone over the threshold to develop symptoms. Next, and so here's just summarized, just summarized that genetic Alzheimer's disease, we tend to think of one gene in the family. Its impact is large. And by contrast, at the other end of the spectrum, we've got the sporadic Alzheimer's disease where many genes are involved, and each of those genes has a small impact. So next slide. So there is one particular gene known as ApoE, abbreviated as APOE, and this gene is involved in both sporadic and multifactorial Alzheimer's disease. So again, going back to that jar analogy, it's the two jars, on the jar on the far right and then the middle jar. So this particular gene comes in 3 alleles. Another way I like to think about it is that these are flavors. These flavors have their names. One is E2, E3, and E4. Only the E4 flavor or E4 allele is associated with increased risk for Alzheimer's disease. And a person can have 0, 1, or 2 copies of E4. You may remember from the pictures that Dr. Robak showed, we get 2 copies of each gene from our parents, one from mom, one from dad. And so this is how a person could have 0 copies, 1 or 2. 0 meaning that right? Neither of your parent had E4 and passed it on to you. 2 copies of E4 would mean that you inherited a copy each from your parents. And so most white Americans have 2 copies of E3. This flavor of ApoE is actually considered neutral. It neither increases nor decreases a person's risk for Alzheimer's disease. Next. And so about 21 to 25% of white Americans has 1 copy of E4. So it's actually not uncommon. A quarter of Americans has this. And much more rare is having 2 copies. So about 2% of white Americans has 2 copies of E4. And we've known for many years that the E4 flavor is a pretty powerful risk factor. So you can see here, this chart represents, at the top, the bar represents a person's risk to develop Alzheimer's disease if they have 2 copies of E4. So that ranges, there's a range, and it ranges from 51 to 68%. The bar below it represents the risk that someone has to develop Alzheimer's in their lifetime with 1 copy, and that's at 18 to 35%. And compare this then to someone who has 0 copies of E4. So that's about 10%, meaning that 1 in 10 individuals with 0 copies of E4 will develop Alzheimer's disease in their lifetime. And you may have heard Dr. Robak discuss this, but lifetime is usually considered into one's 80s. So these numbers were derived from statistical modeling, but we have more current data, next, that suggests, that suggests that actually, if you look at a population that is drawn not just from Alzheimer's research centers, but also drawn from the community, that in fact 2 copies of E4 may confer a risk of Alzheimer's that's closer to 31 to 40% in one's lifetime. So if we take that and think, that still means that 60% of people with 2 copies of E4 will never go on to develop Alzheimer's in their lifetime. Next. And so genetic risk factors among Black and Hispanic Latino Americans are not well understood. We know that Black and Hispanic Latino Americans have a greater lifetime risk for Alzheimer's disease than white Americans. And we also know that the rates of E4 vary among the different populations. Next. A greater proportion of Black Americans has 1 copy of E4, whereas by contrast, a smaller proportion of Hispanic Latino Americans has 1 copy of E4. So generally speaking, ApoE gene testing is not advised. It is available, but it's not advised. There are professional societies that recommend against ApoE testing. This includes the American College of Medical Genetics and the National Society of Genetic Counselors. The test, and this rests on really the fact that the test cannot predict precisely if someone will develop Alzheimer's disease, its risk. There are no ApoE E4 based interventions for now. Next. And it's important to keep in mind that you could have 0 copies of E4 and can still develop Alzheimer's disease. We cannot ignore family history. And next, and so this does, however, raise some issues where, the medical community advises against it, and yet there is great interest in this test. Next, there's some concepts that the medical community uses, notably something called clinical utility, right? How effective is a test at guiding medical management? And this is one test where the medical community would say that it doesn't really change medical management, but that's often pitted against this sense of personal utility, this idea that this is people's information. It is important to individuals for other reasons other than medical management. And so, so certainly an active debate.
And so I think what I'm going to do is advance to the slides on ataxia, Jamie.
Sure. Yeah, so ataxia, I mean, it's a fancy word that basically describes a movement problem affecting balance, coordination, and walking. It's used in a variety of different ways. I think it can be used to describe someone's movement. You know, a person's walk or a person's gait is ataxic. That just, again, describes the movement. But it can also be used to describe a group of disorders known as ataxia conditions or primary ataxia conditions. Ataxia can have many causes. It can be secondary to a different condition, or it can be its own primary problem, where that is the persistent and most pervasive issue, one of movement. Ataxia disorders, again, these are primary ataxia conditions. They're usually associated with a region of the brain known as the cerebellum. So this is a small part of the brain on which the two cortices sit. It's the part of our brain that controls movement. It's the part of the brain that regulates our emotions. Next. And if you've heard a physician refer to what they see on a brain scan, if they're concerned about an ataxic disorder, sometimes they'll describe cerebellar degeneration, or sometimes they'll refer to it as spinocerebellar degeneration. Next. Next. So ataxias can be acquired or they can be degenerative, meaning that they progress over time. They get worse over time. The acquired ones can be reversible, and there are many causes, things like like tumors, infections. There can be nutritional deficiencies, including vitamin E, vitamin B12. Certain medications can cause problems with movement, specifically ataxia, things like lithium and valproic acid. And toxins, alcohol, lead, and mercury can all cause movement problems. And then there's a group of disorders known as the autoimmune conditions that can also lead to ataxia. Next. But with regards to the degenerative or progressive forms of ataxia, these are the types of ataxia that may be more often seen in a neurogenetics clinic. One sporadic type of degenerative ataxia is known as multiple system atrophy. Next. But I think, certainly from a genetics perspective and a genetics talk, there is a focus on spinocerebellar ataxia. These are a group of hereditary degenerative disorders, and where ataxia is the primary progressive problem. Next. It can manifest in a person's walk. People can have an irregular path of movement. They can kind of sway. They can walk as though they are drunk but are not. Next. A person can have a wider stance, known as truncal instability. There can be hand and limb movement problems where a person can have trouble with fine motor skills because they move past an object that they're trying to pick up. Next. And then speaking there's a huge motor component or movement component to speaking. People with spinocerebellar ataxia can have an altered prosody, which is an altered rate of speaking. There can be variable pitch with kind of a motor-like or sing-song pattern. And then there can be difficulty with volume regulation. Next. And there can be also optic nerve problems. Next. Oh, so we won't view that, but there are several more. There are a handful of spinocerebellar ataxias. The most frequently occurring are given kind of designations or names, notably SCA 1, 2, 3, 6, and 7. They tend to be numbered. These particular SCAs are due to what's called a repeat expansion. And so this refers to blocks of DNA or chunks of DNA that are called repeats situated in a gene. And we all have an expected number of blocks that are lined up in tandem, end to end to end. And when a person has a more than usual number of these blocks or repeats, it's called an expansion, and that's when the ataxia can develop. And so people with SCA tend to have at least one parent with the same diagnosis or similar symptoms, and usually there are more relatives. And sometimes the family history can be quite vague. We can simply hear about a relative who had progressive movement problems. They were always clumsy and discoordinated. And that got worse to a point where they needed a cane or a wheelchair to get around. Yeah, and so here's just an illustration. For those of you who enjoyed the visual representation, on the top we have a gene, an SCA gene, and these chunks, these repeats, are represented by the string of letters CAG. And there's just a usual number that we expect in people. And when that number is in excess of what we might expect, that's when that leads to the problems that cause ataxia. I'll turn it back to Laurie now, or Dr.
Our last topic is going to be ALS before we move to the Q&A, or amyotrophic lateral sclerosis. And it's good that we're going back to this because hexanucleotide repeats are involved in ALS. Okay. It's also known as Lou Gehrig's disease for a famous baseball player who was diagnosed with this a while ago. But basically what ALS is, it's a progressive disease that involves the nerve cells in both the brain and the spinal cord. And the primary output is really motor because if you no longer have the neurons that control movement and they die, the brain can no longer initiate and control the muscle movement. And so the problem is with the neurons in the primary motor cortex, which is this hunk of the brain that's outlined in orange, which sends signals down through the spinal cord to the upper motor neurons. And that's what's shown in green. And all this can be affected and result in ALS. I'm showing a picture here of Stephen Hawking, who's someone who's very famous, who had ALS. The early symptoms can include muscle twitches in the arm, leg, or shoulder, or tongue, muscle cramps. The muscles can get tight and stiff, and I think Jamie and I both mentioned spasticity earlier on. You can get weakness affecting any part of the body, really, slurred speech, and then difficulty chewing or swallowing. And as the disease progresses, it kind of moves outward in. And so maybe your hands and feet may be involved initially, but eventually what happens is individuals lose the ability to speak, eat, move, and even breathe. The disease typically lasts about 3 to 5 years, and the cause of death is typically respiratory symptoms because your muscles can no longer allow you to breathe. Although there are some individuals who survive for many years beyond the initial diagnosis, including Stephen Hawking. The symptoms usually start in childhood or late adulthood, but really the average age of diagnosis is in the mid-50s. Males are slightly more affected than females, and it seems to be more common in those of Caucasian or white and Hispanic descent. And just like in Parkinson's disease, about 10 to 15% of individuals with ALS have someone else in their family with ALS. There are 4 famous ALS genes that I'll talk a little bit about. It's a little bit different than what we've been talking about with Alzheimer's disease and Parkinson's disease, because all of these genes, if you have a mutation in one of these genes, you're going to get ALS. And they're dominant, which means that your children would have a 50% chance of inheriting it. Most common is C9orf72, and I'll spend a slide talking about this. And then SOD1, FUS, and TARD-BP are also the genes, and that's from most common to least common. So C9orf72 is the most common genetic change in ALS for those of Caucasian descent. And this, instead of being 3 letters that are repeated over and over, there's 6 letters that are repeated over and over, 2 Gs and 4 Cs. And this just shows you where in the gene those repeats are located up in the upper portion, all those black lines that are close together. And in people with C9orf72, their risk of getting ALS increases with age. So about half will be diagnosed in their mid-50s, and 100% will be diagnosed by age 80. And so as you age, your risk of getting ALS increases if you have this mutation. The age of onset varies based on what gene is causing ALS. So SOD1 causes earlier onset ALS than C9orf72, and SOD1 is more common, of course, than FUS and TRD-BP as well. It's interesting because ALS genes are associated with another neurologic condition, and this could probably be its own evenings with genetics talk in and of itself, called frontotemporal dementia, or FTD, which is a neurodegenerative condition which causes memory issues and issues with behavior and language, but can also cause motor problems in about 30% of people. And interestingly, a lot of the ALS genes also cause FTD. And there can be individuals who have both ALS and FTD. And we know that 3 out of the 4 ALS genes that I talked about are associated with FTD. And SOD1, for reasons that are not entirely clear, is not. And we know for ALS, even though it's billed as a primary motor disease, about 50% will have some sort of cognitive problem over the lifespan of the illness. And so there's some interplay between the two conditions. So if you have ALS, how likely are you to test positive for a gene change? So it really depends on whether you have other family members with the disease. If you have a family history and you yourself have ALS, 60 to 75% chance that you're going to have a gene change in one of these genes that I mentioned. If you don't have a family history, which is known as sporadic, it's going to be less. It's going to be 6 to 10%. And this is just showing you that the genetic variance based on reported ethnicity, whether a person reports themselves as being either European or being Asian. So a person who is of European descent tends to have, and this is, the top is those with the family history and the bottom is those without. But you can see that in those of European descent, C9orf72 is the most common cause, whereas those of Asian descent, it's about 30% SOD1. And so the genetic change really depends on self-reported ethnicity as well. And then in the bottom, you can see for those who don't have a family history, the genetic testing yield is much lower. All that blue there basically means that the cause is unknown. So this is just to kind of wrap up with why you might consider testing for a neurodegenerative condition. So the reason why people tend to come see us in clinic is, the most common is, I really want to know why this happened to me. And we call this therapy diagnosis because sometimes people just really want to have an answer for why in particular this happened to them and not somebody else. It can also help you determine the risk to other family members. There are gene-specific treatments that are quite rare for now, but it's really showing a lot of promise, particularly in the Parkinson's disease field, is the ability to participate in gene-specific clinical trials. There are many advantages and disadvantages, but these are the main ones. The disadvantages can include cost. As I mentioned, insurance may or may not cover these tests and they can be costly. It can cause anxiety knowing that you have a genetic change and that it could potentially be passed on to family members. And the Genetic Non-Discrimination Act is a law that was enacted by Congress that prevents you from losing health insurance if you test positive for genetic condition, but it doesn't protect you against losing other forms of insurance. For example, disability and life insurance are 2 things that you may want to consider getting before you would commit to genetic testing if that was something that was a priority to you. So there are 2 ways to see an adult neurogeneticist. One is the Adult Neurogenetics Clinic at Baylor. It's located on 7200 Cambridge Street. And you can call in order to make an appointment. And then I have the URL here as well. And perhaps Amanda or Susan, we can share this URL either in the chat or email it to the participants as well if they're interested. But you can really just go to the BCM website and just type in adult neurogenetics and you should be able to find us. And then there's also the Consultagene Clinic, and I'm going to turn it over to Jamie to let her talk about that.
Yes, thank you. The Consultagene Clinic is also a service of Baylor College of Medicine, and it is a virtual genetic counseling clinic. We see indications for neurodegenerative disease, including Alzheimer's, Parkinson's, and related conditions. This is a cash pay clinic, and there's a number here at the far right if you want more information. And certainly, we are very connected to the Adult Genetics Clinic and are happy to refer on when appropriate.
And so at this point, we wanted to wrap up and thank you all for your time and attention, and especially your patience with our technical details. And I think we've got we've ended right on time, so we should have a good 15 minutes for Q&A.
Oh, I don't hear Amanda.
Oh, sorry, I was double muted. Thank you to both of our speakers. And I see that Susan just dropped the link to the seminar evaluation in the chat. So if you guys could complete that, that would be much appreciated as well. And then we do have about 15 minutes for Q&A. So if there are questions that people have, please feel free to drop those in the Q&A section, or I'll also give you a chance in just a minute to unmute yourselves as well to ask questions. We do have one I see that's come through about Alzheimer's disease, so I'll give this one to Jamie. Somebody asked about the genetic risk for Alzheimer's disease in African American and Latinx populations. Why are those not quite as well understood? Is it just that there haven't been enough studies in those populations?
Yes, so I think that question, thank you for the question. And I think this really highlights the great need for additional research. You know, I think historically, you know, Hispanic, Latino Americans and Black Americans have not been well represented in Alzheimer's disease research. And there are greater efforts to change that. I think we need to include individuals of all ethnic backgrounds in our Alzheimer's research studies. So thank you for that question.
And Jamie, you have actually a grant studying this very thing, correct?
Yes. Thank you, Dr. Robak. Yeah, I mean, we're conducting a pilot study right now among Spanish-speaking Hispanic Latinos to interview them and ask the words that they use to describe precision medicine, genomic sequencing related to Alzheimer's disease.
Okay, thank you. I have a question that I'll ask Dr. Robak. So both of you guys touched a little bit in your lecture on how genetic and environmental factors can kind of both play a role in these different conditions. And I know on our end, when we talk about pediatric conditions that have a similar mechanism, sometimes families have questions about what exactly environmental factors are, what that means. Could you touch a little bit on that, and maybe in the context of neurodegenerative conditions, maybe what are some of the more common environmental factors that we might think about?
Well, for Parkinson's disease, I think it's fairly easy because we know that there are a couple of neurotoxic substances that directly cause Parkinson's disease. For example, MPTP, which is an impurity that was found in LSD back in the day, selectively kills the neurons that cause Parkinson's disease. We also know that there are pesticides like rotenone that are mitochondrial inhibitors that seem to cause Parkinson's disease. And so it's nice because we have kind of those well-defined environmental factors. I think what's less well known is, you know, what are the different lifestyle modifications that can potentially prevent Parkinson's disease and other movement disorders. And it's a difficult area to study because you have to consider the genetic changes, you have to consider the person's entire lifetime and the entire environment that they've been surrounded in. And so I think we often use the word environmental because it just means we're not sure, even though we do have some well-defined environmental causes for some of these diseases. I don't know if there's anything you wanted to add, Jamie.
Yeah, no, I wanted to say that, you know, I don't think we know of the equivalent toxic exposures in Alzheimer's disease as we do with, say, MPTP and rotenone, paraquat in Parkinson's, but certainly, you know, certainly we can't ignore a person's lifetime kind of environmental exposure.
Okay, thank you. We've had another question come up about Parkinson's disease. So I think this one is for Dr. Robak. Melissa asked, you mentioned a study for genetic testing for Parkinson's disease. Can you let us know where to find more information about this?
Yeah, so there's actually a number of genetic studies going on right now, and it kind of depends on whether you yourself have Parkinson's disease or not. There are many more options if you do. We have one going on right now at Baylor, actually multiple studies going on right now at Baylor, but you have to be a patient of the PDCMDC, or the Parkinson's Disease Center and Movement Disorders Clinic at Baylor. But there are larger genetic efforts going on as well throughout the country, looking at individuals who have risk of Parkinson's disease based on family history, and also individuals who have Parkinson's disease and want to know the genetic basis. And so it really depends on your individual situation as to which direction we would point you. And I'd be happy to, I'll just put my email in the chat. If you want to just email me, I'd be happy to talk further.
Oh, I think we've had, she said it's for a family member.
Okay, great. Just feel free to email me and I'll be happy to point you in the right direction.
Great. That is, for now, all of the questions that I've seen come up in the Q&A. Are there any other questions that anybody would like to either unmute themselves and ask, or any questions that anyone wants to send through the Q&A?
Hi, this is Susan Fernbach, and I have a question with regards to finding out about studies that are ongoing. Would you think that people should go to the specific disorder foundations, or go to the clinicaltrials.gov website, or both, I guess?
Both. For Parkinson's disease, a great resource is going to be the Michael J. Fox Foundation. They are responsible for running a number of different clinical research studies on Parkinson's disease, where you can talk to your local neurologist or neurogeneticist as well. There's really a variety of ways to find out about studies, and there's really no bad way. Jamie, maybe you want to speak to more Alzheimer's disease research?
Sure. Yeah, I mean, clinicaltrials.gov will certainly turn up a lot on Alzheimer's disease, but I'll also put a message or a website in the chat. There's also numerous registries for people who are interested in receiving more information about Alzheimer's breakthroughs and discoveries. And there's this particular one, endalznow.org, which is a passive, easy passive way to hear about discoveries.
Yeah, and thank you for reminding me. I'll put it in the Michael J. Fox Foundation website as well.
Sure, and thank you. And I guess for specific Baylor studies, would they be on the Baylor website that people can look at?
Yeah, sometimes yes and sometimes no. You really have to be seen by a Baylor neurologist, I think, to get into many of the studies that are specific for some of these disorders, though. But we can certainly direct you in the right place, to the right place.
Yeah, I mean, I think there's, I mean, that sounds like a wonderful resource, and maybe that's forthcoming. You know, the BCM does have a new Center for Alzheimer's disease and neurodegenerative diseases, known as CANDY, led by Dr. Josh Schulman, a neurologist with whom Dr. Robak and I work closely. That particular website does not have a list of studies, but it tells a little bit about the research that we do, and maybe in the future we might be able to have kind of a go-to location for studies.
Thank you. This is great.
We've had one more question come up in the chat related to Parkinson's disease. So somebody asked, can lifestyle change minimize Parkinson's disease risk?
So this is always a fraught question because, so one of the things that we actually know is that smoking decreases your risk of getting Parkinson's disease, but of course we would 100% never recommend that you start smoking because that causes other problems. Caffeine consumption can also be a risk factor, but these are all very small effect sizes. And so I don't really think that there's necessarily any lifestyle modification that would really prevent PD, other than the association with smoking, which again, please do not start.
Those are all the questions that I've seen for now. Does anybody have any last questions that they'd like to ask? Oh, we've had another question come through. This one's related to Alzheimer's disease. So somebody mentioned that they have had 2 family members who have developed Alzheimer's. Would you suggest getting a genetic test despite genetic Alzheimer's disease being rare?
Yeah, that's a great question. I mean, certainly I would say that if you have any concerns about your family history and wanted to talk to someone about that, you know, Dr. Robak and I are available. I would encourage perhaps a referral to one of our clinics, you know, certainly because I think there could be kind of a tailored conversation that you might want to have with your provider.
And we've had another question come up. I think you might have touched on this a little bit, Jamie, with one of the previous questions. But any information about, well, actually, so this one's about ALS. So any information about ALS and environmental effects, such as pollution, petrochemical plants, any data about that related to Alzheimer's disease, or I'm sorry, to ALS?
Yeah, I think the data for that is even less well known than it is for Parkinson's and for Alzheimer's disease, and so I really can't speak to that.
I will give it just a second. Any further questions from anybody for Dr. Robak or for Jamie? All right, I think that is everything then. I see that Susan dropped the link to the survey one more time in the chat. So if you haven't taken that yet, please do. And then our next seminar is going to be on Tuesday, November 16th at the same time, 7 p.m. About the impact of COVID-19 on children with rare disorders. So, somebody said thank you, excellent presentation. And yes, I agree, thank you very much. This was a very helpful seminar, and thank you to both of our speakers for participating. And with that, I will let everybody go. Thank you.
Thank you, everyone.
Thank you so much.